PP acting lumbar. Clear-culp pose, fill-fill cycle and cell division explains the orderly sequence of events by which a cell grows, copies its DNA, and divides into daughter cells. The phases of the cell cycle, the stage E, and how meiosis differs. Let's begin. The cell cycle is the ordered series of events a cell goes through between one division and the next, and it has two main phases. Interphase is by far the longest phase, often over 90% of the total cycle time, and it's when the cell grows, carries out its norm, and critically replicates its DNA. Interphase itself has Pierre Chloe Poke Postamenant Pi Aril Philadelphia Philadelphia Rusilip, Phil acting lumpophotogram, Philadelphia of phase where the cell grows and synthesizes proteins and organelles needed for DNA replication. S phase synthesis phase where DNA replication actually occurs, doubling the cell's genetic content, and G2, the second gap phase where the cell continues growing and prepares specifically for division, synthesizing proteins needed for mitosis itself. The M phase or mitotic phase is when actual cell division occurs, encompassing both division of the nucleus, mitosis, and division of the cytoplasm, cytokinesis, producing two genetically identical daughter cells. Cells that have permanently or temporarily stopped dividing, like most mature nerve cells, exit the active cycle into a resting state called G0. Mitosis is divided into four main stages. Prophase begins with chromatin condensing into distinct, visible chromosomes, each already consisting of two identical sister chromatids, joined at the centromere, from the S-phase replication. And the nuclear envelope begins breaking down while the spindle apparatus starts forming. Metaphase is defined by all chromosomes lining UP precisely along the cell's equatorial plane, the metaphase plate, with spindle fibers from opposite poles attached to each chromosome's centromere. This precise alignment is essential for what follows next. Anaphase begins the moment sister chromatids finally separate, pulled apart by the shortening spindle fibers toward opposite poles of the cell, and it's specifically at this exact moment, chromatid separation, that the chromosome number is genuinely doubled, since each separated chromatid now counts as its own independent chromosome. Telophase is essentially the reverse of prophase. Chromosomes decondense back into loose chromatin, the nuclear envelope reforms around each set of chromosomes, and the spindle apparatus breaks down. And cytokinesis, dividing the cytoplasm into two separate daughter cells, typically overlaps with and completes shortly after telophase. Meiosis is a specialized type of division, occurring only in germ cells, that reduces the chromosome number by half, producing genetically variable haploid gametes from a diploid parent cell, and it involves two successive divisions. Meiosis 1, the reductional division, is where the chromosome number is actually halved, and it differs from mitosis in a crucial way. Homologous chromosomes, one from each parent, pair up tightly together during prophase 1 in a process called synapsis, forming a structure called a bivalent or tetrad, and while paired, non-sister chromatids from the two homologous chromosomes can physically exchange segments, a process called crossing over occurring at points called chiasmata, and this exchange creates entirely new, recombinant combinations of alleles on each chromosome, distinct from either original parent chromosome. At the end of meiosis 1, complete homologous chromosome pairs, not sister chromatids, separate from each other, and how each pair of homologous chromosomes orients and separates, is entirely independent of how every other pair does, called independent assortment, and together, crossing over and independent assortment are the two primary sources of genetic variation generated during meiosis. Meiosis 2 closely resembles a normal mitotic division, except starting from haploid cell then diploid 1 crossmer finally separate here just as in mitosis, producing 4 haploid daughter cells in total from the original single diploid cell, each genetically distinct from one another and from the parent cell, due to the crossing over and independent assortment that occurred earlier in meiosis 1. Comparing the two processes directly, mitosis produces two diploid, genetically identical daughter cells, used for growth, tissue repair, and asexual reproduction, and it involves only a single division, with no pairing of homologous chromosomes and no crossing over. Meiosis, in contrast, produces 4 haploid, genetically variable daughter cells, used specifically for sexual reproduction, and it involves two successive divisions. with homologous pairing and crossing over occurring specifically during the first division. This fundamental distinction, mitosis maintaining genetic constancy for growth versus meiosis generating genetic diversity for reproduction, is the single most important, most frequently tested concept in this entire chapter. Let's recap. The cell cycle S interphase, G1S and G2, prepares the cell for division during the M phase. Mitosis proceeds through prophase, metaphase, anaphase, and telophase, producing two identical diploid cells, with chromosome number doubling precisely at anaphase. Meiosis, in contrast, involves two divisions, with homologous chromosome pairing and crossing over in meiosis, one generating genetic variation, and independent assortment adding further diversity, ultimately producing four genetically distinct haploid cells. Remember the core distinction: mitosis maintains genetic constancy for growth, while meiosis generates diversity for sexual reproduction. That's cell cycle and cell division. Keep the mitosis versus meiosis comparison table crystal clear. It's one of the most reliably tested topics in all of biology. See you in the next video.